I'm Lakshmi Amaravadi. Today we'll be talking about post-marketing immunogenicity monitoring. So far you've been learning about safety and efficacy, PK/PD impact of immunogenicity. And now we will look beyond that and look at what are the implications post-approval of a drug. And I will also present a case study that will illustrate some challenges as well as share with you some insights from that. So the learning objectives of this particular portion of the course will be to look at and examine the process of assessing impact of immunogenicity. We will talk about key elements of immunogenicity risk management plan and what are the distinctions between post-marketing commitments versus post-approval monitoring. And I will present a case study to illustrate the following elements. These will include clinically relevant anti-drug antibody classifications, a post-surveillance program that was implemented. What does it mean to develop a commercial immunogenicity assay for post-approval surveillance? Challenges related to managing post-approval assay qualifications across testing laboratories. And then finally, some of the considerations related to designing these surveillance registries. So let's look at what is immunogenicity monitoring. And first and foremost, it is important to distinguish between risk assessment and risk management. I'm sure you've heard about the term risk assessment so far in the earlier chapters of this course. Risk assessment is prospectively planned assessment based on the molecule, the type of the molecule, what is the mechanism of action of that molecule, is it a redundant protein with an endogenous important functionality versus is it a non-redundant protein with an important function, is it a product-related feature that is going to influence immunogenicity, as well as finally the target population. All of these factors influence risk of immunogenicity. Immunogenicity risk assessment should be incorporated into well-designed clinical trials. These impact assessments should include impact of anti-drug antibodies on safety and efficacy. Safety-related impact assessment typically will include risk of infusion reactions due to development of anti-drug antibodies, risk of serious anaphylactoid reactions, and there's always the risk of interacting with an endogenous protein that has a non-redundant function that can lead to serious consequences, such as clinical deficiency syndrome. Although it is not listed here, that as well as a number of other factors come into play when you think about impact on safety. And that one needs to keep in mind always when assessing impact. And coming to efficacy then, compromised efficacy primarily is due to development of neutralizing antibodies, although clearing antibodies can also have an impact because you do not have drug exposure.